better get this type of correct, just so I can tell you what we're gonna do. So we're gonna have a conversation right now with Yoshiro Kano, who is at Colorado State University. All the way from as of last night, flew in from Tokyo, Japan. Currently on sabbatical, hanging out in Chicago. And he's going to share with us some thoughts on range-wide abundance and temperature models. That's then going to lead us into some small group breakouts on that very topic. So let's welcome Yoshiro.
Well, thank you. And can everyone hear me okay online?
Step in here.
I'll step in here. Okay, cool. Yeah. So, okay, assume everyone's okay online. So I have some updates on the southeastern analysis of temperature and trout abundance. And discuss other next step is going to be the range-wide analysis, which I like to get some feedback from you and make it as collaborative as possible. So I'm from yes, Colorado. So that's an interesting place because we hate brook trout. We're trying to kill them for the benefit of cutthroat trout. But I've been working on brook trout in the east coast for about 20 years now, since I did my PhD at University of Connecticut. And I think our goal ultimately is to use data and models to inform prioritization spatially. And I think all of us in this room are interested in the questions, the kind of questions like where do climate refuge exist, and where should the next management action to take place? The biggest bang for the money. And joint venture also has a data and science committee for that reason. And the regional analysis, we're interested in regional analysis because data are available, right? So like many agencies collect trout count data and temperature logger data, but they're often stored locally on someone's local computers. But then when regional analysis happens, it's very insightful. Here's an example of Dave Kazak's range-wide analysis of genetics. So we can actually see the 3 genetic clusters longitudinally, and we also see some evidence of fish being northern fish stocked. Well, this doesn't work. Yeah, like northern fish stocked in the south. So the regional analysis really kind of give us the overall big picture about the resources. And I would like to do a similar range-wide analysis for temperature and count data. So these are the discussion questions I think I'm going to pose again at the end of the slide. And I'm here because I want to make this range-wide analysis as interactive as possible by getting feedback from the group like this, and then providing feedback back. And then these questions were prepared in consultation with Lori, a few others. And then I'm basically relying on the existing data sets, right? So, and I also like to make the product more useful for the trout managers. So that's why I like to kind of have this conversation to lead off the afternoon session. And once again, I'm going to come back to this slide. So in terms of timeline, we've been working on the Southeastern analysis in the last 3 years, and it's funded by the Southeast Task and U.S. Fish and Wildlife Service. And we have received a new multi-state conservation grant from the FWS to work on the range-wide analysis. And our goal is to look at the temperature and abundance data from Georgia all the way to Maine. So the Southeast team, there are some familiar faces. Some of you are here in the room and virtually, but I'd like to kind of give a big shout to Lucy and George, who have done the heavy lifting for the work I'm going to present on the next a few slides. And the big takeaway so far from Southeast analysis is that there's a lot of much spatial heterogeneity in thermal habitat and population responses to climate variation. Okay, and diving into some of the key results, I analyzed paired stream air temperature loggers collected by Andy Doloff at U.S. Forest Service, and he had 203 sites with paired air and stream temperature collected at 30-minute intervals for 5 years. And when we looked at those data, the thermal capacity or the sensitivity was fairly obvious. So these are the weekly mean air temperature and the stream temperature. The stream on the left has a very buffered thermal regime where the temperature regime is fairly stable over time. Whereas the one on the right is a fairly sensitive side where stream temperature responds readily to the air temperature. So when we look at this kind of nonlinear relationship, the next thing we did was to look at the slope of this nonlinear curve. Okay, and so that's the beta right here. And the regression slope, the ecological meaning of that is that it's the increasing stream temperature when the air temperature increases by 1 degree in Celsius. And if we map those regression coefficient or the slopes, we do see some spatial patterns here in the southeast. The beta values are shown in the cooler to warmer colors, with the cooler colors being more buffered. So we do see some thermal refugia farther in the south, which kind of makes sense because they're at the higher altitude or elevations. And there's a lot of variation, but then when we did the PCA analysis of the 174 habitat variables available in the GIS, we could only explain 17% of the variation. So we do know that there's much variability, but we're still kind of doing the poor job of explaining why that spatial variability does exist. But then this work has kind of shown that there are thermal refugia that are likely to occur at the southern latitude where there is high base flow index, that's an index of a groundwater input, and smaller watersheds compared to larger downstream ones, and lower channel gradient, which may be a surrogate of other things like, more wetland, whatnot. And if we can use that patterns and we can actually predict the thermal sensitivity or the regression across all of the some 8,600 NFP plus segments that are available in the southeast. So we do see some clusters of what might be the thermal refugia, but that I'll probably caution that the spatial clusters may partly due to the spatial interpolation, which we have to kind of enforce to make the model kind of look right. And then diving into the trout count, in the past, there has been the presence absence analysis range wide. Mark did that back then. And then, to our knowledge, nobody has really attempted to put together all the abundance data. So we put together abundance data based on multi-pass and single pass estimate of electrofishing. And then we also basically try to describe or account for the different capture probabilities among data sets. And that's how we were able to combine all of the abundance data into a single cohesive analysis. And we have, this is still ongoing, and this is the kind of where I like to get the feedback later on. But so far, we have looked at trout abundance at the HAC10 level. We're now seeing if we can actually go finer into HAC12. But then here on the left, there are the warmer colors indicate higher adult density. On the right, we also looked at the temporal variation in adult density, meaning that if temporal variation is small, these are more stable. The population size is more stable over time. We also looked at the temporal trends as well. And from these different metrics, we can actually come up with kind of multi-dimensional assessment of population status using these abundance data.
Yoichiro,
Yes.
could you go back again once? Can you just repeat what you said about what the variation indicated, which direction it went?
Yes, so on the left, it's the abundance, so mean abundance. Warmer colors indicate higher adult density, so more fish. And then on the right, warmer colors indicate higher temporal variation. So cooler colors means more stable population size over time. Does that?
Yeah.
Perfect.
Your time, when you say time, does that mean within a season or is that
No,
over?
multiple, over years, among years.
Okay,
Mhm. Yeah, so these are all kind of annual data.
Wait, you got a question from Mike?
Yes.
What time, so you mentioned 1,000 sites and
Mhm.
temporal scale and lots of map. What is kind of the earliest?
Yeah, the 1982,
And then up to 2023 or?
up to like 2000, like 19, 18. Yeah. Yeah. Yes, Mark.
Did you account, was that trout abundance or brook trout?
Yes, brook trout abundance, yes.
So did you account for if it was an allopatric or sympatric population?
No, we did not.
With the temperatures of air?
We could. Yeah, I know there's like Chesapeake Bay group is actually, I mean, you did it too,
Yeah.
like looking at the allopatric, sympatric combinations of rainbow, brook trout. We haven't done it yet.
Yeah, I was just thinking with exotics, as brook trout may be less stable,
Mhm.
even with temperature, they would have to have that variable in there.
Right, right.
It'll be interesting because, you
Yeah.
know, like some of Andy's data sets, and
Uh-huh.
when we had at Fridley where we sampled 25 years in a row,
Right.
the power to
Mhm.
detect differences, the coefficient of variation is so high
Right.
on young in the year and adults that
Mhm.
you can barely tell a 50-60% change in population
Right.
statistically. So
Right.
this is just incorporated by the total numbers is how you're getting this model.
Yeah, but I mean, so I'm kind of cutting out all the details, but it's a Bayesian hierarchical model. So we can actually borrow individual site information to come up with like HAC 10, HAC 12 level assessment. So borrowing the individual data into a hierarchy can kind of give us more confidence in our estimate. But you're right. I mean, inherently at the segment level, site level, there's a high temporal variation.
Yeah.
You know, CV is like typically over 100%, right? So there's higher standard deviation compared to the mean. And it's worse in younger fish than
Oh, yeah.
adult fish. Yeah, but still, like, you know, we're kind of trying to do a finer resolution of abundance prediction. So this is at the NHD plus segment scale. I mean, we could interpolate or we could come up with a model, but then how precise is it, or how confident can we be at doing this type of thing? So kind of like to come to this spatial resolution that makes sense to the managers too, right? So that's why I'm kind of showing this slide as a food for thought. Okay. And we also looked at some spatial population synchrony. So here's the last kind of a piece of information analytically I'm going to present. So spatial population synchrony refers to similarity in temporal trends among sites. And I'm going to visually show that on the next few slides. But then asynchrony kind of generates portfolio effect, makes the regional resources in aggregate more stable over time. And then, you know, perhaps one approach for planning landscape conservation is to identify a suite of robust populations, you know, along that synchrony gradient. I mean, just like, you know, coming up with a portfolio of populations at the broad spatial scale. So explaining the population synchrony, visually, you're going to see a time series of trout count at the single site. So here, population fluctuates over time, and then this can be explained as a trout count as a function of time. Okay, and then I'm going to go to a multiple sites now. Here, you're seeing 3 sites, and then they're in parallel, so they're in complete sync, right? So we can actually explain the trout count as a function of time and site here. But then in reality, we see kind of a spaghetti plot like this. But then statistically, we can actually incorporate the time and site interaction to explain the degree of spaghettiness, if that's the word. Yeah.
I'm gonna use it.
Yeah. Thank you. Yeah. So if that's okay, spaghettiness is high. That variability here should be high. So we can actually calculate ICC or the intra-class correlation coefficient, where if it's very spaghetti-like, right, the ICC, the denominator will be large. So ICC will be small. So on the next slide, I'm going to show you the distributions of ICC, where 0 is asynchronous and then 1 is synchronous. And then many of those sites have ICC values less than 0.5, meaning that we have a fairly messy spaghetti plot in this area. So no matter how we look at it, there's a lot of heterogeneity over space in terms of abundance, mean abundance, synchrony, temporal trends, temporal variation, and stream temperature as well. So, and I'm going to provide more details or technical details in about a month when I give a webinar for the Southeast task. So that's on November 29th. And then if you're interested in, I think you can register free of charge from that ctask.ncsu.edu. So that's partly why I didn't really cover the details today. I want to kind of give a big, kind of bigger overall picture. And now towards the range-wide analysis. Like, so we're building upon the experience in the South to expand the geographic scope to all the way to Maine. And also, I like to use this opportunity to further increase collaboration with scientists in the Northeast. So, Jim Fair, Ben Litcher, they have the Northeast grant to look at stream temperature and the flow for the Northeast. And I mean, I can't separate with Ben because he was on my PhD committee and he was my postdoc advisor. And they also have this great data visualization tool called, what was the name for that? ICE, yes, right. And then it's a great way to communicate the data or the findings with a wider range of audience. So eventually what we're going to do is to use a different matrix. It's like similar to like IBI, right? So we use a different matrix to characterize or rank whatever the spatial resolution, whether it's the HAT12 or the NHD plus or the catchments, to identify areas which are most likely to support brook trout in the warming climate, or where the restoration may have the biggest benefit for the population persistence. And I would like to do it in a collaborative research, in a collaborative way, where the trout managers contribute data, provide management relevant questions, and then we provide the models and the visualization tools, perhaps discuss the sampling protocols by identifying areas where you might need more data. And then I wrote the multi-state conservation grant in a very general way. So it's very wide open as to what exactly I'm going to do. So I really want the product to be more management relevant. So I think with that, that concludes my presentation. And then I can finish, or like I can leave this slide so that we can break into the groups. Yes. But then basically these are questions related to what scientific tools might be helpful in terms of temperature and abundance models. And then maybe that's a segue for a broader science data needs that EBT or joint venture may where we may want to bring up, or like each agency might need.
Okay. So both in the room and on Zoom, please note these two questions. I only had one of them, I think, on my next slide. So capture both of them, because this is the conversation we'd now like you to have in breakout groups. So as long as one member of your breakout group has captured it, we're good. So please grab it on Zoom, write it down in the room. So if you're taking photos of it, that's perfect. And Yoshiro, thank you very much. We're going to move into breakout mode here. Let's just do a little quick transition. If you could log off from there and actually just log off from Zoom altogether.
Oh, okay.